Okay, so here's our course. Remember, under Files, under Unit 3, we have our objectives and our PowerPoints. So we're doing Innate first, and I have it in 2 PowerPoints. Then we'll move on to Adaptive, which I have 2 PowerPoints for, and then we'll do Disorders last. And I'm hoping to do this immunology activity. So we have a lot to do before the test. And where I have it scheduled right now, I might actually move it. I can't remember why I have it that day that I have it, so I'm working on changing that. So the other thing I wanted to point out to you guys is last time, under Pages, you'll see we have web pages, animations, and videos for Unit 3. And I'll be updating this. But these are the 2 web pages for homework that I wanted you to, for that assignment, to look at. Maybe play the game if you wanted to. Last time we watched this video, this YouTube video. Again, a pretty good overview of the immune system. And then we watched some of these movies, also linked up here. We watched all these movies, actually. So I'm going to be referring to them today, and so if you missed that, they're here for you to go over. So I just wanted to point those out, and I think I even mentioned them in my PowerPoints. So for the first one, we're going to go over phagocytosis and inflammation, which were the focus of those movies that we watched last time, those short movies. So, right, we watched these videos that are on that page I just showed you, the leukocyte rolling, we watched the leukocyte homing, right? So this is all related to the inflammatory response. So we actually watched it in action last time. So for our objective, we really want to outline, right? What are the steps that happen? We really want to focus too on the factors that initiate the response. So why does this start? And what are the outcomes? What happens once inflammation is started? So this is an animation that I have embedded in my PowerPoint, but I will also add these, and in fact, come to think of it, I think I already have added a file, but I'll add them as hyperlinks later for you guys. I'll show you. In the files, there is, under Unit 3, probably a document. Yes, immunology. It's a document. So these are all, here's even those links to those YouTubes, but then here's all these animation links. And so sometimes, too, it actually has a little quiz associated with it, and it says you can email your instructor. Do not email me. You can take the little quizzes, that's fine, but I don't want your results, right? The results, as far as quizzes and stuff, are your Connect assignment, which I hope you're remembering to do, right? And I did not factor into your guys' midterm grades. I'm actually going to do a real, calculation this weekend. And those of you guys that are struggling and worried at whether you're going to get the grade you want in the course before potentially dropping on Tuesday, I'll have that information for you guys on sometime this weekend. So come Monday, you can call or come by or discuss if you need to where you're at. So all these links are here. I don't know that they're hyperlinked though. So here's that inflammatory response one. I have to keep allowing this thing. So there it is. So I just have it embedded in my PowerPoint. And instead of listening to the person, so in PowerPoint you just have to be in slideshow mode for these buttons to actually work. But I'm going to mute the person so I don't have them talking in my ears. But you also notice it's closed caption to tell you everything that they're saying as well. But I'm going to kind of add my own audio. So we kind of watched this last time in a similar animation and some actual movies of happening in real animals' bodies. So this shows white blood cells, and specifically ones we're going to talk about, neutrophils, later, going through a capillary or a vein, and see an injury has happened, and that injury may have even let invaders like bacteria shown here. When that happens, these cells are sending out signals, right, because they've been damaged. Other cells also may be detecting the presence of these foreign substances, these bacteria. Resident cells like mast cells, they can detect and they can release chemicals that are going to cause changes to the endothelial layer, the layer of the blood vessel. And then these white blood cells also have receptors on their surface for the receptors that are going to become available on these endothelial cells. And the whole reason for this is to get the movement of these cells into this area, right? So it's all chemical communication and the use of receptors and molecules that they can actually bind to. So when that happens, and so one of the major things that's released by, one of the major chemicals by mast cells is histamine. And what that does, again, it's going to create the environment such that these cells are going to be attracted and stick and even roll along the surface and be able to head out from the blood vessel into the tissue area where damage has happened. The other thing that is going to leak out of these vessels when they become permeable is fluid. So that's why when we experience inflammation, what are the things that you see? We talked about this last time. When something's inflamed, it's swollen. And again, it's that fluid leaking out of the blood vessels when these white blood cells are also traveling through the area. What's the other thing you see or feel? It's warm. And again, blood flow to that particular area is going to increase, right? Your capillary beds aren't always full in particular areas, but they will open up those blood vessels and they'll really rush as much blood to that area as possible to get as many of these white blood cells to the area as possible, right? So with inflammation, that redness is the increased blood flow, as well as the heat you experience. Your blood has most of the heat that's associated with your body, right? So the increased blood flow is the redness and the heat. The fluid attributes to the expansion that happens in that area that puts pressure on the pain receptor, so that's also why you'll feel pain sometimes. And then additionally, sometimes you'll have loss of function because it impedes movement. So one of the things that you'll also see here, this particular type of white blood cell has an ability called phagocytosis. So it's literally gobbling up, you see that? The bacteria, ingesting them and destroying them. And the particular white blood cell we're looking at is a neutrophil. And the reason why I know that is because they get called during inflammation, right? That's the only time they actually leave our blood and go into the tissue. And I can also tell by the way they drew these cells. So if you take AMP2 lab, you actually have to identify these cells under the microscope. So under the microscope, these cells have a very unique nucleus. It's actually multi-lobed. See how it has multiple sections to it? And they usually have cytoplasmic granules, but they're not very distinct from their cytoplasm. So sometimes you'll see little dots, right, but they almost blend in. And so these guys are called neutrophils, neutro referring to neutral staining granules. And I always like to think of fill as being filled with granules, right? That's how I remember all the granulocytes, right? All their names end in fill. And we'll talk about those guys. So inflammation. Any area where you have injury, okay? So in this case, again, something puncturing into your skin. That damages your cells. And then if it allows entry, and it can even be your normal flora that are on your skin, right, that get pushed deep in, that can then invade into the tissue. And then you're going to have other eaters, and we call these guys the big eaters, macrophage, right, for big eaters. These are always found in your tissues, and there are even specific ones in all of your organs, and they're kind of your guard dogs, right? They're always present, and so if something gets past, breaches that very important barrier of, say, your skin or your mucous membranes for your intestinal tract into the tissue, then these guys can eat them up right there on site. But in addition, they can send out chemical signals to call more cells to the area. Not to mention any mast cells are in the area if they become activated, or your cells themselves. So notice again these chemicals, pro-inflammatory chemicals. So, you know, you've got cells that are injured, you have white blood cells that are on scene that recognize there's an invasion. They're going to send out these signals, and they're basically calling for backup, right? And so you'll get this increased blood flow, right, to get more white blood cells to the area. You're going to get neutrophils heading to the area, and as I said, they only travel into the tissue when we have inflammation, when they're called there. You're going to have additional macrophages that are going to head to the area. They're called monocytes when they're in your blood, and then they differentiate to the cell type we know as a macrophage when they're in your tissues. Again, big eaters. So one of the other videos, right, diapetesis actually means oozing, right? So this is that movement of the white blood cells squeezing out of the capillaries into the tissue. And they literally interact, right, to receptors on the surface of the endothelial cells that are only present when inflammation has been initiated. This is going to trigger them to move into that area. So tissue damage results in inflammation. The purpose of it is to make sure it's a localized response, and we even watch that in those videos, right? The cells are you're just going to where the injury happened. We want to eliminate any invaders that have gotten in, and we of course want to restore the tissue function. What happens during this process? Well, they're byproducts of what's going on, right? The swelling is the leaking out of the blood vessels. The redness is the increased blood flow and that heat, because blood is hot. Pain, again, the swelling pushing up against the pain receptors. And as I said, sometimes loss of function. Well, how does this whole thing get started? Well, your cells themselves have receptors on their surface to be able to detect when things go wrong. And we generally refer to these as pattern recognition receptors. So your body recognizes specific patterns that are related to pathogens. So this is pathogen-associated molecular patterns. They love these acronyms. It's the one thing I'm not so fond of in microbiology. The good news for you guys is I'm not going to expect you to memorize them. D, danger-associated molecular patterns. The important thing is, right, it's patterns we're recognizing. How are we doing that? We have specific receptors. Right, so some examples of some receptors are the toll-like receptors. And the I forget what the NL are. See? Even I forget. So the good news for you guys, not a graduate level immunology course, right? We don't need to know the specific receptors and what they're doing, right? We just need to know that they exist and their general purpose for existing. So they're going to detect, right? These receptors are going to allow us to detect when something has gotten in that's bad news. How do they know? They recognize general patterns that are associated with the bad guys. with these pathogens. And then they're going to release a signal. For our cells, it's all a chemical signal, right? They don't yell like us. They don't have voices, right? They're not a skunk. They're not going to spray. They're going to send out chemicals, though, right, that are going to be able to bind to receptors on other cells and get them to trigger them to do something. Generally, we refer to these chemical signals as cytokines. They're almost like the hormones of your immune system. Just like hormones are the chemical signals of your endocrine system, cytokines are the chemical signals of your immune system. Histamine is the one most people are familiar with, right? So this is a specific chemical signal, a cytokine, released by certain cells in your body and causes other cells to mount a specific response to that signal. So the microbes themselves, right, can induce the release of cytokines, right, trigger this inflammatory response, as well as tissue damage when the cells are damaged. When blood vessels are damaged, two enzymatic cascades are activated. One that's going to lead to coagulation and one that's going to lead to increased permeability. Now, of course, when the blood vessel itself is breached, we don't want uncontrolled leakage into our tissues, so that's why it coagulates. The second reason, too, is to be able to stop organisms from using that superhighway to get to other areas, right, to move out of the area where the infection has occurred. We don't want them hopping on to the blood vessel superhighway, right, because in a matter of a minute it could travel the whole distance of your body, and that's at resting heart rate. That's not even when you're all hyped up. But we do have to have increased permeability, right, in some areas so those white blood cells can get to the area, eat them up, right, and destroy any pathogens that may have gotten in. So it's a cascade of events, right? We're going to get dilation of those blood vessels in the area. Those capillary beds are going to fill up, those venules. And again, as I said, that blood flow, increased blood flow, is actually going to be why it's hot and red. Even though the blood flow is going to be increased to that area, because of the dilation of the blood vessels, it'll be at a slow rate. Right? So it's not moving very fast as compared to under normal conditions. And under those conditions, of course, the leaking of the fluids results in the swelling and the pain. What is truly happening, though, is that those white blood cells, those leukocytes, are grabbing on to those endothelial cells. This will slow them down, allow them to squeeze through, right, diapedesis through the blood vessel into the area. And these are phagocytic cells. These are cells that can engulf the bacteria and literally destroy them. Clotting factors are going to wall off the site of infection, right? We want to try and localize it to a particular area. Dead neutrophils, tissue debris will accumulate. This is what we know as pus. And the bad news with pus is that not only does it contain our own cells and dead white blood cells, it could also contain what? Bacteria, if you have an infection, right? Viruses too, right? So it could contain pathogens. And that's why, of course, you want to not expose that to other parts of your body or to yourself. You want to be clean when cleaning up pus. Acute inflammation is short-term inflammation, and the macrophages are usually able to clean up damage by ingesting even our own dead cells and debris. If acute fails, if acute inflammation fails, then we go into what's referred to as chronic inflammation. And this is basically when acute has the inflammation is not taking care of the problem, right? Those white blood cells coming to the area isn't enough to eliminate the problem. So we get cells still coming to the area, they're not able to eliminate the invader, right? We're going to need our backup, our adaptive immune that we'll talk about next. So some things that will happen sometimes when we're not able to eliminate them is the white blood cells are still going to keep coming, the macrophages are going to come, and they'll accumulate, and they can even fuse together to form giant cells. Or granulomas, and granulomas are usually the macrophages with the addition of T cells coming to the area. This happens very often in tuberculosis, an infection in your lungs. The mycobacteria tuberculosis is engulfed by the macrophages, but the macrophages can't kill it. And so then the T cells start trying to get involved, and they just keep accumulating and trying to keep the pathogen in. The good news for us is the slow grower, right? So this accumulation happens over time, okay? The good news nowadays is what do we have to treat tuberculosis? Because it's a bacterial infection. Antibiotics. The bad news is we got a lot of resistance going on. And the reason for that is because you've got these bacteria inside of your cells, right? So treatment with antibiotics is 6 months to a year, if not longer. You know how many people actually keep up with that long of a treatment on a regular basis? Not too many. So then, we get this buildup of resistant bacteria in the environment. So we have strains that are multi-drug and even extremely multi-drug resistant strains of tuberculosis. And that's the scary part. And again, it's slow progressing, so people don't realize they're sick, right? And then they're really sick, and it's really progressed. Back in the day, they used to refer to this as consumption, right? So there's lots of famous people that are believed to have died from consumption. One of the most common ones that people are aware of is good old Doc Holliday in the Westerns. They believe that he died of tuberculosis, of consumption. And again, over time, because they didn't, have antibiotics or didn't know they could treat it with antibiotics, it would just overtake the lungs, and the people wouldn't be able to breathe. They would basically suffocate to death. Right? Even though the immune system is trying to do the best it can, it's not taking care of the problem. Make sense? There's lots of infections where, again, our cells are just going to keep coming to the area and try and contain it, right? Keep it there and try for it to not go anywhere else. But that, in its essence, could create problems, right? Especially where that inflammation is happening. If it's happening in your lungs, eventually over time, you're going to run out of space to breathe. If it's happening in your brain, it can cause destruction to your brain cells and death. So what happens when we have inflammation? The bad news with this is that once you elicit this, it's been likened to fire sprinkler system, right? So once it goes off, right, everything gets wet. It's not, what's the word I'm looking for? It's not specific, right? So if the fire alarm goes off and we have sprinklers in the building, the computer lab is toast, right? Because computers are not big fans of fire. But we'd probably be able to save the building, right? But the contents would be seriously damaged, especially when you're talking about things like computers. So this is kind of what happens in your body, too, is that when these cells go into this area, sometimes in the effort to kill the bad guys, they'll even kill some of our own healthy cells. It's like bombing the whole building to kill one guy. Any innocent bystanders get killed as well. So enzymes, toxic compounds from our phagocytic cells are released and damage tissues. And because of that, as I said, neutrophils only go to the areas into our tissues when they're being called there under the inflammation. I equate these guys to our suicide bombers, because they literally will kill themselves in an effort of trying to kill the bad guys. So remember how we talked about eukaryotic cells have a quite complex cytoskeleton? We've actually found out that neutrophils will shoot their cytoskeleton out of their cells to create like a net to try and trap the bacteria in a particular area. So they literally kill themselves to try and create a net to trap the bacteria in a particular area. Also, when they degranulate, if they release their granules to the outside of the cells, they die in that process. So again, they're just dropping their bombs, and they're not running away. They're going to die on scene. And they have really toxic substances in their granules. If limited, right? So if you say have a cut on your finger, the damage is usually minimal, right? You're not going to have a lot of damage by your own white blood cells trying to clean up the mess. But delicate systems like your membranes that surround your brain and your spinal cord, what we refer to as your meninges, when things like that become inflamed, it can be very serious and life-threatening. And the additional problem with that is those are very important components of our body. So what are they encased in that helps protect them? Your spinal cord and your brain. Bone. Bone. That's kind of hard stuff, right? Unlike our skin, what does it do when we swell? It stretches, right? Bone doesn't stretch, right? So your vertebrae, right? Your skull. When you got inflammation and buildup of fluid there, it just pushes back on the brain and the spinal cord, right? And so, again, that can create more problem than be helpful. So cell death in the inflammatory process. So as I said, when cells get damaged, right, it's going to elicit inflammation. Well, your cells actually die all the time. Right? They get used up, especially your red blood cells, man. They're long distance truckers, right? They're running on that blood superhighway all the time. About every 120 days, they retire for good and get replaced. They're pretty beat up. So that process of them being removed and parts of them being recycled, that happens in such a way that it doesn't elicit inflammation, because we would be in a constant state of inflammation all the time in our bodies, especially with the amount of red blood cells that get deteriorated over time. So there's a special process called apoptosis, and this is programmed cell death. And this does not trigger the inflammatory response. So this is when the body's like, okay, you've retired, it's time for you to go be recycled. We don't need to get in an uproar. We don't need to sound the alarm. Cryopatesis, on the other hand, if we recognize invasion, if those pattern recognitions are triggered, then the cell may undergo cell death with inflammation. So again, like say if a cell gets infected by a virus, gets infected by a bacteria, if we get triggering of that sensory system, that alarm going off, then we are going to have inflammation, because there's something else going on here other than just normal cell death. And sometimes too, though, if there may be something going on with the cell, the immune system may have that cell die and not elicit inflammation. It'll go through apoptosis, or what we call programmed cell death or cell suicide. Like if you go on your peaceful way, like you're supposed to, we won't call in the cavalry, right? But if you don't behave, we'll call in the cavalry after you. So how do our cells actually kill and digest foreign invaders? This process is referred to as phagocytosis. And not all your white blood cells have this ability. And some do this a little bit differently and do additional things than just eat them up and spit them out. So the two main eaters are your macrophages and your neutrophils. And so again, last time we watched these videos of them moving and grabbing onto bacteria. Now we're going to look at the stepwise process right down to the molecular level of what occurs at each step of phagocytosis. And then we're going to look at the similarities and differences between macrophages and neutrophils. So for phagocytosis, we have, for instance, a pathogen like a bacteria. You'll have a cell, a phagocytic cell, whether it be a macrophage or a neutrophil. It can sense this foreign invader by chemicals that it releases just normally. This guy can recognize it and head towards it. These cells move like amoeba. And then the very first thing it needs to be able to do is be able to grab onto it or basically attach. So here they're showing you that one of our systems that we're going to talk about next is the complement system. These are specialized proteins that can bind to the surface of bacteria. So one of those is called C3b. And I always remember B for binding, because that's what it does, right? So when it attaches to the surface, it like marks this bacteria even more so for destruction by phagocytic cells. Because phagocytic cells, like the neutrophils and the macrophages, those big eaters, is they have receptors on their surface for these types of molecules. And then again, they have those pattern molecules. So they recognize generally bacteria, the bad guys. And in this case, a protein in our body recognized it and coded it for us. So we've tagged it for destruction. So the very first thing that needs to happen with these phagocytic cells is they need to come to the area. All right, and they're going to be called there during inflammation, especially if they're a neutrophil. Or you may have a resident macrophage that senses it and he's going to head towards the pathogen. The next step is they literally have to attach to it, be able to grab onto it. So they have to have their receptors on their surface, be able to grab onto something on this pathogen. And that's going to initiate the next step of the process, where they're literally going to bring their membrane around the pathogen and surround them in membrane, forming what we refer to as a vesicle or a vacuole. When it's phagocytosis, we give it the special name of being called a phagosome. I was trying to remember if they were going to put both the words there. Nope. Okay. So then once the phagosome is formed, and so we've trapped them, right, in basically a little cage, a little vesicle called the phagosome. And then phagocytic cells have lysosomes, right? Remember, these are vesicles that contain what? Enzymes, digestive enzymes, right? So these are going to fuse with the phagosome. And guess what we do with the name? We fuse the name. It becomes a phagolysosome. When you have the fusion of the lysosome with the phagosome. And those enzymes are released, right? And we start digesting this pathogen. Oh, there we go. Phagolysosome. So it's destroyed. And then you're going to exocytose, dump out the little bits. Now what they don't show you here is that cells like the macrophage and the dendritic cells that we'll talk about later actually pick up pieces and present them on their surface in a process called antigen presentation. And this allows them to communicate with other cells of our immune system like T cells. Neutrophils don't do this. They just eat. So stepwise, chemotaxis refers to that movement to the area. And one of the compounds that's present in our bodies that helps movement is a protein, again, part of the complement system we'll talk about later, is C5a. So it's actually this phagocytic cell that's sensing that chemical and is heading towards these microbes. In addition, the complement protein has coated them, this C3b. So as you can see, again, these guys have receptors. So they're able to attach, grab on to that pathogen, and engulf it into that vesicle we call a phagosome. The lysosomes fuse, we got a phagolysosome. We digest, we destroy, and we exocytose, spit the extra bits out. That's all the steps of phagocytosis. Right? The phagocytic cells have the ability to do. Gobble up, eat, and destroy. So how are these two major phagocytic cells similar and different? Macrophages are always in your tissues. Right? So you have those resident macrophages on guard all the time. Neutrophils, on the other hand, only travel into your tissues during inflammation, when they're called to the area. If neutrophils degranulate, if they release their granules, they will die. Macrophages can survive after phagocytosis. They can eat and eat and eat. Macro meaning large. Phago referring to eating. Neutrophils, as I said, these are the big time killers. These guys have the greatest killing power. And they're even willing to kill themselves to kill the bad guys. So those nets that I talked about, right? They'll actually literally shoot their cytoplasm out, their cytoskeleton, excuse me, out of their cells to create these nets to trap them. So my nickname for neutrophils is suicide bombers. They're serious about killing. They'll even kill themselves to take the job done. Macrophage is a little more sophisticated, right? And sometimes they need help from their friends. So they can be what's called activated by T cells. And these will help increase their killing power. So it's like they kind of consult with their friends, are like, I don't know about this guy, right? I'm having trouble eliminating him. Any suggestions? They're like, well, why don't you try this other bomb or chemical to try and kill them, right? Or I've seen this one before, this is what works. And as I said, sometimes when they can't kill the invader, they're going to fuse together and form giant cells. And when the T cells get involved in forming those masses, we refer to them as granulomas. And this specifically happens, granuloma happens, formation happens in the lungs with tuberculosis. So we're going to take a break, and we'll come back and we're going to look at the different white blood cells and talk about their functionality. So 15 minutes.